to achieve a normal facial appearance. So when we look at the face, we all know during the early embryonic stage development, the neural plant cells migrate into the ventral part of the developing embryo and help to contribute to various facial structures. And when you look at the anatomy with the well-formed face, and then under the skin, so there are muscle cells, and then also bone and cartilage, and then nerve and blood vessels, all these structures together help to build a beautiful face. So really this highlights the importance for us to understand what are the cellular sources that contribute to the various parts of the craniofacial structure, and also the genetic the gene expression patterns that correlates to these structures development. And also when we look at the craniofacial development, we also must appreciate the fact that the formation of mandible, maxilla, and tongue and palate are very much related. So if you look at the developmental stage from E10.5 all the way to E15.5, and then looking through a coronal section, and then you notice the development of the mandible and the tongue and the palatal shelf. So really, as the development continues, these structures have such a close relationship. And one of the syndrome was mentioned earlier, Pierre-Robin sequence, in which you have a small mandible that interfere with the descendants of tongue, and that may cause cleft palate. So in that case, the palate is actually it's abnormal, but it's a defect of the mandible that leads to the defect in cleft palate formation. So the point is, we have to look at these structures together and see how they integrate in this process. So our goal is really to look at the specific gene expression and couple with the 3D imaging analysis during the development of the mandible and maxilla. And then we're looking at, we're using microarray RNA-seq, and also highlighting some of the important gene expressions by using in situ analysis, and I'll show you some of the data there, and also 3D imaging coupled with precision measurement, and to look at the development of mandible and maxilla. And our goal is really to enable hypothesis-driven research and into the regulatory mechanism of mandible maxilla development, and also generate information to be used as a blueprint for craniofacial tissue regeneration. And these are sort of the milestones that we have listed over the course of 5 years for our project. And as you can see, for the control, we have about 60 microarray data set. And then when we get to the mutant model, I'll show you some of them. We are generating these microarray gene expression data. And then also with the in situ analysis and cell lineage tracing and 3D imaging. And then at the end of my presentation, I want to show you a video, hopefully that will play, and then to sort of give you an idea how we wanted to integrate all of this data together, so someone can just go to a mouse embryo and click and slice, you'll be able to find all these information at one location. So these are the data sets that we have submitted, and then there the microarray data at Geo, and then so all the controls have been completed, and that the mutant, TGF-beta mutant models are in the process of being curated. So what we have been doing is to looking at the distal region or the proximal region of mandible, or the distal region of maxilla and proximal region of maxilla, to compare the gene expression in these different domains. There are multiple studies published before indicating that before we see the different morphological phenotypes in this area, there are genes that help to control, to set up these kind of subdomains that later on leads to the development of a body portion of the mandible versus the ramus part of the mandible development in the distal versus the proximal region, respectively. So what's the quality of our data? That's something that we really pay a lot of attention, and we have been running a lot of this analysis with the help of Vasu and Paul Thomas at USC. So here, looking at the differentially expressed genes between the distal region and the proximal region of mandible, and showing you 3 samples from each region, and you can see they segregate quite well, and then highly expressed versus underexpressed genes in the different regions, and then with a heat map analysis. And then you can, from there, look at some of these significant pathways that are reached in the distal region versus the proximal part of the mandible development at E10.5. And so when we look at these top differentially expressed genes, we always are very pleased to see these genes that we picked up correlate with what has been published in the literature. So here you can see at the lower right corner, for example, the HAM2 gene is expressed in the distal region of the mandible, and versus the DLX 1, 2, and 3 are more in the proximal region. So here represented in the negative value. And then so these other genes on top of this list are, most of them are not known in this process. So that really gives us a sort of a very important list for anyone who's interested in patterning of the mandible maxilla development to go on and do this type of study. So we have also been working on developing software in which people can go to the FaceBase hub and then pick our data that from the different part of mandible or maxilla, and then perform this type of differential gene expression analysis. And then from there, that will generate a list for you. And then here I'm highlighting the top 20 differentially expressed genes between the distal region of mandible versus maxilla. And then here I pick goosefoid, which is really a well-known gene to be involved in regulating mandible development. And when we learn that, and then we come back to either whole mount in situ or section in situ analysis, I'm always really pleased to see data like this. Here you can see the expression goosefoid in the control sample, and then here's one of our mutant, the Wnt1 L5 mutant, where the goosefoid expression is downregulated. And you can see the data is really quite clean, looking at LHX6 expression in the oral domain in the mandible, and then versus in the mutant. So this gives you really an idea, not only using the gene expression analysis, you can see where the genes are, the ones that are being picked up by this analysis, then coupled with these kind of detailed gene expression map, if you will. So we have really been working very hard to map these genes into the different domain within the branchial arches, in this case, at the early stage of mandible development. And we can clearly separate the mandible into the distal region and also the proximal region, and then you divide this into the oral and aboral. And then these are the different genes expressed in the different domain. That gives you an idea of how the genetic sort of blueprint, if you will, that help to set up these type of development. And from there, this last year we have generated a lot of these gene expression maps. So these are not just real data, but a sort of a schematic drawing. And the one you can go on to look at this, and then look at where BMP4 is expressed, in the maxilla mesenchyme and in the mandibular epithelium, and then with the detailed description and also publication or using our published data to support the drawing of this type of gene expression pattern. And then here another gene, important gene, LHX6, in the different stages of facial development. P63, another one, and then a patch. So you get the idea that how this type of data coupled with the gene expression analysis can help someone to look at the importance of the function of these genes in regulating craniofacial development. So that's gene expression. And then cell fate. We have published this a few years ago looking at the contribution of neopress-derived cells using the Wnt1 cream crossed with the Rosa reporter generated in Phil Soriano's lab. And then here using the U5, looking at the monogenic-derived cells in the first arch and the epithelium. And then here you also have a map showing what are the cellular contributions to the facial skeleton at newborn. But clearly, there's a gap between these two. So there are multiple stages that are not present. So in the past year, we have been filling that gap, looking at the cell fate in the different stages of development. So all of that now is available at the hub. So you can see the contribution of the different cellular components within the craniofacial structure coupled with the SEM images. So you get to see how these cells contribute to the various parts of craniofacial development. And the last part of our study is really these 3D imaging analysis, looking at when mandible and maxilla began to form, and then how they really take on these anatomical form, and then give rise to this really well-defined anatomical structure and throughout the developmental stages. And then we have added anatomical landmarks for us to really define these different parts, the distal region and the proximal region. And this gives us the opportunity to really analyze these structures in more detail. So here, giving you an example, in control, you can see the mandible, maxilla, and premaxilla. And then in the type 2 receptor mutant, you can see the mandible is basically smaller, but both the distal and proximal region are still present. But when you look at L5 mutant in this example, virtually the entire proximal region is gone. So that's something, in the past, what we have relied on is either microCT or de-staining, so you get to sort of do a tracing and then get the sense, oh, this proximal region is really affected in this mutant model. But we argue that is really not enough. We need to be able to give this type of more precise measurement, developing these 3D viewers software, so allow you to pick a mandible and add these anatomical landmarks, and then so you can measure the distance in any two, between any two given anatomical landmarks. So that way you have a sense, what is really the norm. And to our surprise, one of the things that we have done this year, we call it deep phenotyping. In the past, we just said, oh, when you have a heterozygous mutant in the L5 mutant, really everything is normal. The mandible looks just as in the control. But is that really true? So when we begin to analyze these anatomical landmarks, for example, if you look from number 1 to number 9, that's from distal to the proximal where the condyle is, and you can see there's already a reduction within the heterozygous model of the L5 mutant. And then, so this type of analysis really adds another dimension to our analysis to look at these mutant models, so how we can really get more information about the importance of the signaling pathway in regulating the development of mandible and maxilla. So this is so far what we have provided to the hub, and then all of these are now available for us to use. And then the last part, which Steve, I wanted to show this so you get to see what we have done with our cellular proliferation heat map analysis. This was a project originally supposed to be done elsewhere, but finally we just got tired of waiting and we decided to carry out this study ourselves with the help of Seth Ruffin. So now you can get to see, we actually get to render the whole embryo basically translucent. So you can see where proliferating activity in the pre-migratory neuropress and also neuropress that already migrated into the facial region. And later on, as these mandible begin to form, you can see the condensation of these cells proliferating more, and then also in the tooth development area. So this is a composite of multiple sections, and we're really pleased that this has worked out. And in terms of collaborations, we've been working closely with the hub and looking at the curating the data, and also we did a comparison analysis between our microarray data with data generated in Trevor Williams' group, and then we're glad to report really the quality of data really supports each other, so it's very nice. And then with Joanna's group, we're actually looking at some of the epigenetic landscape comparison between the cranial neuropress and bone marrow-derived stem cells in terms of their ability for tissue regeneration. With Axel, looking at enhancers and some of the cellular activities during development, and we hope we can develop additional collaborations and then interactions with other spoke projects, and then through the interaction here and also during the poster session. So this last one is a movie that took a few months to produce, but I hope the sound will.
The goal of our project is to provide multiple complementary perspectives on the development of the mandible and maxilla in normally and abnormally developing mice. Here is a 3D model of a developing mouse at embryonic day 12.5. We've mapped the fate of different cells that contribute to the mandible, maxilla, and surrounding structures. Here you can see the epithelial lineage, the cranial-neural crest cell lineage, and the muscle. By merging these cell lineage maps, we get a comprehensive view of the distribution of different cell types within a developing craniofacial structure. We have dissected the proximal and distal portions of the mandible and maxilla separately, so that you can use microarrays to identify differences in gene expression in these domains. For example, you can see here the different expression patterns of FoxC2 and HAN2 at embryonic day 12.5. Combining our 3D microCT images, cell fate, and gene expression datasets will allow for unprecedented understanding of craniofacial development in the mouse. Other FaceBase spoke projects, which contribute information about genetics and epigenetics in mice and other organisms, complement ours and allow for an even more comprehensive view.
So what I'm hoping is to use this for two purposes. One is this, as it says, can be used as a video abstract that can be linked to the spoke project, so you can highlight the type of data being generated in that particular project. And then the second purpose for this, something like this, is we already uploaded this video to YouTube, so others can use this to publicize the data that we have, and then to invite users to come to our site in order for them to get familiar with the type of data we have, and then get more users to use the data we have. So with that, these are the people, as I mentioned at the beginning of my talk, and we have really worked very hard. Yuan and Jill is not here. She just gave birth to a beautiful baby, and did a lot of these schematic drawings. And then Vu and Zoe who are all here, and then Bridget, and then Alan, who's just graduated from college, and then waiting to start middle school, and then did all those heat map analysis for us, and then created, with the help of Seth, made a video available. So you know, I learned a lot from this when you are with something that really someone else promised it, to get it done. It didn't work. It pushes you to develop something on your own. And in this case, it really worked out. And I'm very happy to share that with you. I'll be happy to take any questions. Thank you.
Okay.
Speak very close to the microphone. So just a question, clarification about the cell proliferation data. I was a little confused because when you look at the whole mount, it looks like it's rather superficial. But when you also had some sections, and those were in depth, so it doesn't look like it was a penetration issue. How did you actually do it? How are you doing this? Can you give us a little bit more detail?
Oh, so the embryos were cleared, and then these are Ki67 cell proliferation activity, and then we cleared the embryo and then did OPT image with the help of a sense buffer at USC. And we have a poster we can actually give you more details.
Is it possible that while you clear, that's maybe while you clear is all the internal tissues where Phil was noting that you see less proliferation, would the clearing process maybe alter that?
Yeah, so what we're doing is now we're actually doing sections of the E10.5 to just to make sure they still match with what we see in the whole mount, or if we lost any signal. So that's something that we're checking right now.
I was very interested in the beautiful cell lineage tracking you show. Are you planning to also look at lineages like endothelium or nerves, neural cells, innervation?
Yeah, so the answer is yes. We just started crossing the adenine-1 tree with the reporter, so that targets the sensory nerve. So that's something, because in the trigeminal nerve is an important component for the craniofacial complex. So we're doing that right now. We also have some endothelial cell lineage analysis, and then these are still ongoing projects. But so far, what we have completed are the epithelial cell lineage study, the neural crest, and also the muscle cell lineage. And these are the three that we propose in our grant, and the additional ones that are still ongoing.
Future grants.
Yeah.
Joan in the back. So I was really interested in the high resolution CT data you're getting on your heterozygotes, and particularly in reference to some of Benedict's recent work where, at least with the FGF pathway, it seems like a major portion of what these signal pathways are doing is buffering variability. And so I was wondering if you see a lot of more variability in your heterozygotes, or if that's something that's peculiar to what Benedict is looking at.
Yeah. So in this case, we compare control with the heterozygous ALK5 mutant. For those of you who know, ALK5 is the type 1 receptor for TGF-beta. And then we also compare the TGF-beta type 2 receptor heterozygous. In that case, all the TGF-beta type 2 heterozygous mutant, there's no phenotype variation at all. Only the ALK5. And if you recall, the ALK5 mutant in comparison to the type 2 receptor mutant has a much more severe craniofacial phenotype. So we think there is something in this case, really the tissue is very sensitive to the level of ALK5s being present versus the type 2 receptor is less of an issue. So to just go back to your question, it's not just a variation per se, because in the type 2 receptor heterozygous mutant, there is no change in comparison to control.
Anybody else? Okay, thank you, Jan.
Thank you, Chris. Everything works.